Hello everyone. This video is going to review transcription and translation before we take our test this week. So remember that the big ideas were that we make proteins through transcription and translation. Together they make protein synthesis. And the central dogma of biology is that DNA becomes RNA and the RNA becomes the protein. That's the way that we make the proteins. So in the nucleus, the DNA gets copied into mRNA, and that's the part that we call transcription. Once the mRNA is made, it leaves the nucleus, and it's read in groups of 3. Those groups of 3 are called codons, the 3 letters, and each codon matches an amino acid. So during translation, the codons are read, and we get an amino acid chain, and that becomes our protein. So the central dogma, DNA is the template. We have to copy DNA into a form that can leave the nucleus, that's our RNA, and then RNA gets copied, it gets read and turned into those amino acids, and the amino acid chain becomes the proteins. So during transcription, we go from DNA to RNA. So transcription goes over this first arrow. Translation occurs when we go from RNA to a protein. In particular, we're dealing with mRNA. So the mRNA is the one that gets transcribed and translated. So once again, transcription is DNA to mRNA. Think that we're copying the DNA code down. Transcribing, copying. Translation, we're translating the code from nucleic acid language into protein language. Proteins aren't made up of the same basic unit as nucleic acids are. DNA and RNA, their basic unit is a nucleotide. The protein's basic unit is an amino acid. So we have to make sure that we translate everything so that way we make our protein properly. So does this happen in all of our DNA? No, it only happens to the genes. Remember, genes were the sequences of the DNA code that make a protein. The parts that don't make a protein are called non-coding DNA. And non-coding DNA is important because it often ends up controlling transcription and translation. And also it does things like protect from mutations. So what is RNA? It is the messenger that helps carry the DNA code so it can become a protein. DNA can't leave the nucleus. That's why we have to use RNA. So RNA is very similar to DNA, except it's single-stranded, so it's not the double helix, it's a single helix. It has ribose sugar instead of deoxyribose, that's how they get their names. And instead of a T, RNA contains a U, so it has a uracil base instead of a thymine base. And there's 3 types of RNA. mRNA, or the messenger RNA, that's going to copy the DNA and take the message out of the nucleus. The tRNA, or the transfer RNA, that's the one that delivers the amino acids. Think of it as transferring the amino acids around. And the rRNA, or ribosomal RNA, is the one that makes up the ribosome. That one kind of makes sense. So what happens in transcription? The what is that we're taking DNA and we're copying it into mRNA form. Where does this happen? In the nucleus. Why? Well, because DNA can't leave the nucleus. We need to turn it into a form that can leave. And who does this? RNA polymerase is the enzyme that ends up doing the transcribing. It's very similar to DNA helicase and DNA polymerase. It'll unwind the DNA and copy it. But instead of copying it in the DNA form, it copies it as RNA. Once it hits the end of the gene, it'll break off and our mRNA will be able to leave the nucleus. So how do we translate that code from mRNA into protein? How is it going to end up happening? Well, we take our mRNA and we read it in groups of 3 letters. So in this picture here, AUG is our first group of 3 letters, our first codon. Our second codon is GUC. So you read those, and each of them stands for a particular amino acid. So the tRNA is going to read this codon and know which amino acid to drop off. And it does that. We know which ones match up, by using these two wheels. So you have the wheel and the old school chart that's a little bit harder to read. So in this case, let's say we have A, U, G. That's our codon. Well, in order to read it on the wheel, you start with the biggest letter first. So we have an A as our first letter. Then our next letter, we're going to move into the second ring and read a G. Oh, nope, sorry. A U. I'm getting a little ahead of myself. So an A, U is the second letter, and G is our last letter. So we're going to find the G, and once you go through all three of those, it should give you your amino acid. In this case, methionine. We usually end up abbreviating these by using the first three letters. So let's figure out how to do it on the old school chart. So you want to find the first letter on this side. Our first letter is an A. So we know that we're going to be somewhere in this row. Now we have to find our second letter. So we come up to the top. Our second letter is a U. So if we have A and U, we know that we're going to end up being in this box here. Our third letter, you can do it one of two ways on this chart. First, you can come over here and find the G, and that's the row that you're going to be in, so you can drag it all the way across to find the appropriate amino acid. Or once you know the box, you can just kind of read it in the box. You can look through these four and find AUG, and you know that it's methionine. So those are the two ways that we end up reading our amino acids. So in translation, that's what's going to be happening. The mRNA is going to turn into protein through that process of reading those codons and matching it with the appropriate amino acid. And this happens in the cytoplasm or the rough ER, basically wherever there are ribosomes, because ribosomes are the organelles that are going to be translating the mRNA code. And this happens in three steps: initiation, elongation, and termination. They kind of end up making sense when you go through them. Their names make sense. So in initiation, this is where the mRNA leaves the nucleus and binds to a ribosome. The first tRNA is going to bind only at the start codon, AUG. I use that one to help get it in your head. AUG is a very special codon. It's the one and only start codon. So your protein won't even be made unless it has that start codon. Once the ribosome finds that codon, the tRNA is going to come and bind, and it's carrying that methionine amino acid that'll get the whole thing rolling. In elongation, the tRNA keeps bringing amino acids as we go through and read the code on the mRNA. The mRNA and the tRNA know how to match up because of tRNA anticodons. So if the codon is AUG, write it over here, then the way that the amino acid knows to bond to that particular place is because the tRNA anticodon will be the opposite, UAC. So now the tRNA knows where to go, and it knows exactly what amino acid to drop off. The amino acids end up joining together through special bonds. They're called peptide bonds. Think of it as P for protein. Protein bonds, peptide bonds. And in the last step, termination, this is where the ribosome will reach one of our three stop codons. So we only had one start codon, AUG, which codes for methionine, but we have three stop codons. So still not too many, but a lot more than the start codon. UAA, UAG, and UGA, those are our three stop codons. There's no amino acid that ends up matching these, but once we get to a stop codon, the ribosome and the mRNA know that it's time to break apart because the protein is done. So, in our DNA, we have genes. These are the sequences of DNA that end up being turned into proteins. So we read our DNA strand, it gets transcribed into mRNA. The mRNA is read in groups of three called codons, And those codons stand for amino acids. When the amino acids are joined together using peptide bonds, they form proteins. And that's the basic idea of transcription and translation. We make proteins through transcription and translation. The central dogma is that DNA becomes RNA and the RNA becomes the protein. In the nucleus, the DNA is copied into mRNA during transcription. The mRNA leaves the nucleus. It's read in groups of 3 called codons, and each codon matches an amino acid. During translation, that code becomes a chain of amino acids, and that is our protein. If you guys have any questions, be sure to email me, and I hope you study hard for your test.